Okay, बच्चा लोग, welcome back. See, this is a brand new topic and very important. ठीक है? Topic is processes of recombinant DNA technology. Fine. ये biotechnology का एक very important part है. So therefore, focus on this. And a difficult part है बाबू. It's not very simple, and it has plenty of processes, plenty of concepts. And jehetu it is a booming topic, our weightage in NEET examination is also too much. At least 5 to 6 questions, matlab 24 to 30 marks you can gain here. So इस पे focus कीजिएगा, ठीक है? Oh, now look at this. In the processes of recombinant DNA technology, first of all, what do you mean by recombinant DNA or rDNA? Recombinant DNA व rDNA होता क्या है? What exactly is recombinant DNA? Now, in the original DNA, suppose we make certain changes, means insert another specific gene sequence in the original DNA. For example, we need a particular type of DNA which is capable of performing an additional function. It can be a correction, it can be correction of an error. Suppose gene DNA re gote gene sequence achi joda ki gote protein tiyari kochi that is causing problems. Okay, so suppose I have to correct the problem. कौन करिबा? तो डीएनए रू सही जीन सीक्वेंस को आइडेंटिफाई करिके बाहर कर दबा. ता बदल रे गोटे बेनिफिशियल जिनसटी जीन सीक्वेंस री ऐड कर दे परिबा. So this is the process which is referred to as recombinant DNA technology, and the DNA which is obtained is known as recombinant DNA. ठीक है? Fine. तो एटा हला तमोरा recombinant DNA. Recombinant काहे को आगला? Because here we have added a new gene sequence to the original. So this becomes a hybrid, hence recombinant. ठीक है? अरे बुझिएला रे, इज दैट अंडरस्टूड? क्या रे कुछ बोलते नहीं हो? इज दैट क्लियर? बुझ पालो समझते? यस, चलो अच्छा है. नाउ व्हाट आर द वेरियस स्टेप्स इन्वॉल्व्ड इन दिस पर्टिकुलर प्रोसेसेस ऑफ रिकॉम्बिनेंट DNA टेक्नोलॉजी? हाउ? First one, isolation of the genetic material. That particular genetic material or the DNA in which the change has to be implemented. सेटाकु प्रथमे identify करिबा and ताकु isolate करिबा बा separate करिबा. Okay, all these are main headings. Under रे पुनी और several steps are there. Means isolation खाली ऐसे तो नहीं बन जाएगा. ए भाई isolate हो जा. That is not possible. You'll have to perform certain steps in order to achieve this thing. Understood? तो पहले क्या क्या चीजें हमको करनी है, अचीव करनी है, our targets, ठीक है? That we have to find out. Objectives. From those objectives, उसके बाद आएगा steps in order to achieve those objectives. Got the point? So these here are basically processes or objectives. ठीक है? देखो, first isolation of the genetic material. Yes. उसके बाद second one, cutting of the DNA at specific locations, at those places or sites where we'll have to insert that particular gene or gene sequence, उनको आप काट के अलग करेंगे. मुझे लगा cutting or removal of the DNA at specific locations. ठीक. Then number 3, amplification of gene of interest using PCR. जो जीन आपको इंसर्ट करनी है, that is a singular gene. Yes, Suppose our experiment बेलकु हमें प्रथम experiment successful है जीवा बोली माने नहीं. That gene may or may not be successfully implanted. Therefore, यदि हमें गोटिए ही नहीं थिबा, it will be a problem for us. Why? Because पूर्ण तरह previous two steps repeat करिया पड़िबो. So what do we do? हमें सही particular gene जोटा की obtain करिचे तार multiple copies तैयारी कर दवा. So amplification basically implies preparing multiple copies of that particular gene using the PCR by polymerase chain reaction. Polymerase chain reaction. अरे बुझिला रे, PCR basically a machinery है जिससे आप multiple copies बना सकते हैं. That is amplification. Okay. आगुरु এই জিনিসটি কোন করা হতো না, ব্যাকটেরিয়া সাহায্যে করা হতো. But now artificial PCRs are there, and by using it we can amplify it to our need plus even faster. Bacteria normally replicate करिया पे 20 minutes का टाइम दरकार हो, but ये faster है उससे. ठीक है, and also can provide us our requirements within a small stipulated time, as well as bacteria जेहेतु गोटे living entity तार कामटी may not be that efficient as compared to this particular artificial PCR. ठीक है जी. हो उसके बाद insertion of the recombinant DNA into host cell. एक बार आपने multiple genes बना लिए, तापरे ताकू insert कर दलो बा ligate कर दलो कोठे रे original DNA रे. से original DNA रे जो थिला, now it has become a recombinant DNA. A recombinant DNA को again we have to plant into that particular host. So that is what is telling insertion of recombinant DNA into the host cell. ठीक है, so that we can see how does this recombinant DNA actually perform inside the host cell. Okay. उसके बाद next, obtaining the foreign gene product. Our basic objective here is we need to find out what does the recombinant DNA do, what are the various products it can manufacture by synthesize. Okay, so we'll allow that particular recombinant DNA to perform inside the host cell, and after that we'll obtain the product which it has manufactured. That is a part of the biotechnology. Yes. Why? Because biotechnology definition to monothimo, that is use of the cell or its components or its products. Okay. Fine. Cell raw components के बाद cell raw products. That is referred to as biotechnology. तो वही चीज है यहाँ पे. Then finally, जैसा जिनसे theory है गला, we need to obtain it. We need to obtain it. Say जो process of obtaining roila, that is referred to as downstream processing. तो यही steps गुड़ी को, यही objectives गुड़ी को देखी दिया उतरे. पहला है isolation of genetic material from the DNA. ठीक है. Cutting of DNA at specific sites. Fine. Amplification of that gene of interest from by using PCR. तापर insertion करी की making or manufacturing of the recombinant DNA. ठीक है. And then inserting that recombinant DNA into the host cell. Okay. Finally, obtaining the foreign gene product, and lastly, downstream processing, that is extracting the product and putting it under certain steps like filtration, okay, removal of impurities, and so that it can be finally ready for usage. Bujela, that is known as downstream processing. Clear? Hikochi, is this understood, bacha? Sabu jinsa buji parlo? Is this clear? Okay. Okay, fine. फिर देखते हैं. How is this isolation of the genetic material done? Look into it. Now, basically, all those substances which we can easily find around us, that is used. देखो, we can treat bacterial cells. We can treat bacterial cells. We can treat fungal cells, plants, animals, or any other tissues. Keମିତି? For example, the bacterial cells ଅଛନ୍ତି. Remember, bacteria hella, ba fungi hella, ba plants hella. ଏମାନଙ୍କ case ରେ କଣ ଅଛି ରେ ବାବୁ? They have a cell wall. बैक्टीरिया की सेल वॉल याद है? पेप्टिडोग्लाइकैन. फंजाई की काइटिन. प्लांट्स रो सेल्यूलोज. ओनली एनिमल सेल्स रे सेल वॉल नथा है. रेस्ट इन तीनों में होता है. ठीक है. तो प्रथमे हमको से सेल वॉल टी रिमूव करिया पड़िबो. अदरवाइज हाउ विल यू रीच द न्यूक्लियर मटेरियल? तो बैक्टीरिया केस रे पेप्टिडोग्लाइकन रहिला. आकु रिमूव करिया पे कोन यूज करिवा? लाइसोजाइम. यस. ओके. देन फंजाई काइटिन अछि, विल यूज काइटिनेज. प्लांट्स के केस में सेल्यूलेज. बुझला? एनिमल्स ପାଇଁ କି ଦରକାର ନାହିଁ because there is no cell wall. So प्रथमे cell wall remove करिबा and will enter inside the cell. So basically ये पहला step है. उसके बाद क्या करेंगे? After the cell wall is removed, then we will take them and use a mortar and pestle mechanism to grind them into the smaller fragments or particles. ठीक है? The cell is broken, releasing DNA and other macromolecules like RNA, proteins, polysaccharides, and lipids. ठीक अछि? Is this clear again? So basically, enzyme action, and then by using an ultracentrifuge or mortar and pestle, this is soft tissue like liver, etc. You can easily obtain them. ठीक है? Now, the RNA is removed. कैसे remove करेंगे? By using RNAs. RNA can be removed by using RNAs. ठीक है? That is ribonuclease. ठीक है? Proteins can be removed by using protease. If you remember, molecular basis of inheritance rate. Molecular basis of inheritance rate. Avery, MacLeod, McCarty. Avery, MacLeod, and McCarty. On board chire. Avery, MacLeod, and McCarty. Chemical basis of inheritance rate. The experiment they performed to use enzymes to obtain the genetic material. One पढ़ला. तो same जिनिस. RNA ପାଇଁ କି RNAs use करिबा, protein ପାଇଁ protease use करिबा. ठीक अछि? Fine. Done. Okay. उसके बाद क्या? Other molecules are removed by appropriate treatments, like for carbohydrates we can use amylases. For lipids we can use lipases. So RNA by ribonuclease, proteins by protease. Semti sugars by ki amylase, sugars by ki amylase, lipids, fats by lipase. So remove gala. But DNA amu darkar, so DNA is use karibani. Besides DNA, बाकी सब use करके remove कर दबा. Okay? ठीक है. ना उसके बाद once age insert ही गला था मैंने आउ RNA नहीं कि प्रोटीन नहीं कि शुगर नहीं कि लिपिड्स ना थी केवल जहाँ बंची जाई ची से हला तमरो बाबू DNA. एबे DNA कौन करिबा? When chilled ethanol is added The purified DNA precipitates out as fine threads. And gote glass rod ya kiछि भी जिंस यूज करिकी आमे आकु बाहर करि परिबा extract करि परिबा. This mechanism of separation of DNA by addition of chilled ethanol and allowing it to sediment is known as spooling. Remember the term spooling. ये सबुठु easy and convenient method. जोतरे किछि बेसी complications दरकार नहीं. ठीक है, simply chill ethanol add कलो, ठीक अछि, and ता जोगु तमरो DNA टी settle हे गला and condense है कि thread like structures हे गला. तमे ताको धीरे से by using a glass rod or any kind of spatula बाहर कर दी पालो, obtain. ठीक है, understood? So for this thing we have now isolated the DNA. We have isolated the DNA from the cell. ठीक है? Oh. Once the DNA is extracted, next step कौन हमारा असला? We need to cut the DNA at specific sequences. We'll have to cut the DNA at specific locations. How we do that? कैसे करेंगे? Now, purified DNA is incubated with the restriction enzymes. देखो ठीक. Purified DNA means जोटा में spooling द्वारा obtain करे. That is purified DNA because it does not have RNA, does not have proteins, does not have carbohydrates or lipids. केवल DNA अछि. बाकी गुड़िक जिनस हमें remove कर देइछे by using enzymes. Yes. So a जिनसटी जो हमें obtain कले, that is purified form of DNA, it is incubated with, मतलब added with the restriction enzymes. ठीक है. Now these restriction enzymes which you see here, we are talking of which restriction enzymes primarily? Endonucleases. Or restriction endonucleases. काहे की? Only those sites जोठी आमर cutting is required, means sequences that we need to obtain. Only say sequence गुड़िक बाहर करिबा. Rest are not essential for us. Therefore, we refer to them as restriction endonucleases by restriction enzymes. ठीक है? Fine. So as a result, कौन DNA is digested. DNA fragments are separated by a technique called gel electrophoresis. Now, very, very important. What exactly is gel electrophoresis? ठीक है? तो DNA fragments हमें theory कर देले. Okay. Now these fragments are to be separated out. For that purpose, हमें कौन use करचे ना gel electrophoresis technique use करचे. एबे इतने हमें कौरिबा कौन ध्यान दिए. That particular fragments is inside a pool, means solution. So fragments along with a medium must be there. ये जो medium की हमें कहूचे, that medium is basically what medium? ना agarose gel. Agarose gel. Why? Agarose go to obtain होची रे बाबू. Yes. Seaweed रो obtain हला. ठीक है. कौन सी seaweed है normally? Yes. Gracilaria. Gelidium. These are the common seaweeds from which we obtain agarose. ठीक है, a sticky kind of gel-like substance. Fine. तो agarose gel रे से fragments गुड़िको हमें rock कीजिए. ये ने fragments गुड़िको. Okay. Now these fragments along with agarose gel, ठीक हो ची, ता संगे आउ कौन मिसेबो ना इथिडियम ब्रोमाइड मिसेबो। ठीक है, इथिडियम ब्रोमाइड क्या डालेंगे? इथिडियम ब्रोमाइड एंड इथिडियम ब्रोमाइड पके देकी ना मिक्स कर देकी रखी देलु बा अलाउ करलु टू सेटल डाउन। ओके, after this the entire mixture is taken and this is the gel electrophoresis machinery. It house the tray ta, this is the tray। एथेरे कौन है ना हमें सकू पके देले ऐड कर देले केमती ऐड करिवा एई जो जिंसटी देखुछ these are known as wells। so we add the mixture into these wells। ठीक है, ता भीतर पके देलो and once they are filled up and our mixture is emptied तापरे आमे कौन करिबा ना सर्किट टी क्लोज कर दबा। empty। fine, one thing you must know that the DNA is negatively charged, so the fragments are also negatively charged। yes, so they will always move towards the opposite charge। negative जेहेतु सेमाने कुआडो मूव करिवे टू द पॉजिटिव चार्ज। यस, तो सेमाने धीरे धीरे मूव करिया लागिले कुआडे टुवर्ड्स द पॉजिटिव इलेक्ट्रोड। so therefore this is the anode, this is the cathode। ठीक है, फाइन। so negatively charged fragments start moving towards the anode। ठीक है, that is the positive, negative। ठीक है, now simple, इसमें दो concepts involved। one, the negative charge of the DNA fragments, the second is the size। so concept is जितने छोटिया fragment ही होबो बा molecular weight जितने कम होबो से दूर को जाई परिबो। understood? so the DNA fragment जो move करूचंदी, ठीक अछि, the movement is due to the negative charge and gets attracted towards the positive charge। plus केते दूर को move करिवे but distance they will cover, that is due to the size or molecular weight of the DNA fragments। so जो DNA fragments छोटो थिबे से माने दूर को जिवे, जो गुडी को बड़ थिबे से माने पाखे रहि जिवे। बुझ पालो? ओके, नाउ ये हो गया आपका basically the gel electrophoresis। now once this is done and fragments गुडी को ता हिसाब रे aligned हे गले, you can see here largest towards the cathode, smallest towards the anode। clear? तो एई हिसाब रे fragments गुडी को सब तांकर पोजीशन हिसाब रे अलग अलग अरेंज हे गले। एबे inside that particular table, ए टेबल टी को अरे जेल इलेक्ट्रोफोरेसिस टेबल टी को आमे के समय छाड़ दवा काई छाड़ीबा बिकॉज़ एगारोस जेल जोटा अछि वो आपकी लिक्विड फॉर्म में अभी ताको हमें अलाउ करिबा टू सेटल डाउन सो दैट इट विल फॉर्म अ जेली लाइक सब्सटेंस, मतलब सेमी लिक्विड काइंड। तो सीए यदि ना तमे नॉर्मली जेली एटसेट्रा घरे खाई थीबो इन ताकुल होता है वैसे ही इट विल बी जेली लाइक। फाइन, नाउ क्या कॉन्सेप्ट लगा इधर भाई देखो, सेम थिंग आई हैव बीन टेलिंग यू। द डीएनए इज नेगेटिवली चार्ज्ड So it moves towards the anode. Okay. The DNA fragments are separated according to their size. Very important. Remember, question exam में पूछा. This question was in fact asked. If the separation of DNA fragments is due to, पहला ऑप्शन दिया ड्यू टू द नेगेटिव चार्ज, ठीक है. दूसरा ऑप्शन दिया ड्यू टू द डिफरेंट साइजेस, ठीक है, फाइन. एंड तीसरा रीजन दिया बोथ, चौथा रीजन दिया नन. आंसर क्या होगा? यस, आंसर इज साइज, नॉट द नेगेटिव चार्ज. व्हाई? बिकॉज नेगेटिव चार्ज जो सेमानकर मूवमेंट है, सेपरेशन नहीं हुआ. सेपरेशन है कौन पाई? बिकॉज ऑफ द साइजेस. जहार साइज छोटो रहा से दूर बाटू गला, अलग हो गला. जिनका बड़ा है साइज वो अलग हो गए. So because of the differential sizes or molecular weights, they are separated. So it is the size which is a more important factor in this process rather than the negative charge. Okay, remember this. Okay. So age insert को कौन ना दिया गला? Sieving effect. कहाँ receiving effect? agarose gel receiving effect. काई? agarose gel medium रही ये कुड़ी को अच्छा दी. So the movement of the आपकी DNA fragments, the movement of the DNA fragments is through the agarose gel. So age insert you got a sieve through the pass color. That is sieving effect. ठीक है? Fine. So that is polymer of seaweeds. Now smaller size fragments move farther. ठीक है? The process is repeated with the vector DNA. Vector DNA माने कौन करे? That particular DNA which will carry the gene of interest. The vector DNA का कोई बात? That particular DNA which will carry the gene of interest into the host. ବୁଝିପାରିଲ? ତମେ ଗୋଟିଏ ଜାଗାରୁ ତମେ fragment ନେଇକି ଆସିଲ, ठीक अच्छी. एई फ्रैगमेंट को तो पुणी होस्ट भीतर छाड़िया पड़िबो. अदरवाइज काम जो करियार अछि हमर वो काम कैसे होगा? So how would I transfer this particular gene of interest of mine to that host? से तो आप पे नहीं की, ऐसा तो नहीं है ना भाई, इसका घर उधर है, तुमने नॉक किया डोर, ले बेटा पार्सल डिलीवरी. नॉट पॉसिबल दैट वे. सो वो कैसे करना है? We'll have to insert this particular gene of interest. Now, insert Korea pai, we need to provide similar but same kind of DNA that will act as a carrier or vector into that particular host cell. So, आमे प्रथमे से gene of interest को आने की यही particular DNA रे add करिबा. तापरे total setup को नहीं की सही particular host cell रे छाड़ी दबा. That is how insertion is done. ठीक है? बुझिए लारी? Okay. Okay. Then एथिडियम ब्रोमाइड जो हमें मिस रही थी, वो किस काम में आता है? ध्यान दो. डीएनए फ्रैगमेंट्स तुम्हें सेपरेट कर दो अगरोस जेल रे. ओके, तादरे एथिडियम ब्रोमाइड मिस्टी. ना एथिडियम ब्रोमाइड when ethidium bromide is exposed to ultraviolet radiation, that is UV radiation, ethidium bromide को हमें expose कर ले to ultraviolet radiation, that results in a form of bright रेड और ऑरेंज, ठीक है, बैंड्स और अपीयरेंसेस, ठीक है, एंड हेंस वी कैन इजीली नाउ आइडेंटिफाई और सी कि को जगह गुडी करे आमर से फ्रैगमेंट्स गुडी को अच्छन, ठीक है, समझ में आया? So easily we can now identify the location of those fragments. Yes, that is the primary purpose why ethidium bromide was added to this particular process. Okay? Why? Because ethidium bromide, when exposed to the ultraviolet radiation, results in the appearance in the form of bands of red or bright orange. Okay? अरे बुझेला? It's a very simple thing. Fine. अब उसके बाद क्या हुआ, ध्यान से देखो पिक्चर में. once the bands are formed, then appear करे देखाई गले, एबे हमें ताकू रिमूव करियो, एक्सट्रैक्ट करियो. ओके, कैसे एक्सट्रैक्ट करेंगे? यस, केमिति एक्सट्रैक्ट करियो को ना. once एटा हला तमर से जो टेबल टी जेल इलेक्ट्रोफोरेसिस रो, जेल इलेक्ट्रोफोरेसिस की ये टेबल है. now in that gel electrophoresis table already the bright red or orange bands are there. Simply by using a small scalpel like equipment, we can easily cut out that particular sequence, okay, and then add it to excise DNA band, okay, add it to the gel dissolver buffer. काहे की? बिकॉज उसमें आपकी एगरोस जेल ऑलरेडी लगी हुई है, तो आई डू नॉट नीड द एगरोस जेल, आई नीड द डीएनए. तो यहां देखिए आप, इस प्रकार से हमने इसको काट के रिमूव कर दिया, एम्प्टी से पूरा सेगमेंट को हमें बाहर कर दे, ले काटी की बाहर कर दे, क्यों बढ़िया. नाउ वी ऐड दिस टू दिस डिसॉल्वर बफर, एंड डिसॉल्वर बफर ऐड को ले कौन है गला. Yes, the agarose gel is removed. The agarose gel is removed, and only the DNA is left out. The agarose gel buffer ru kon hela, agarose gel remove he gala, and only kon rahi gala, only the DNA fragment is left out. So amaro kamo he gala, जो जिनसटा हमें चाहूथिले buffer, I mean the DNA fragment that we have obtained. ये जो process टी हमें कॉले, that is cutting out of the desired fragment, ठीक है, from the table and obtaining that gene of interest or the DNA fragment. ये जो process टी करा गला, इसका नाम क्या दिया गया? It is called elution. क्या नाम दिया गया? Elution. ठीक है, this is known as elution. Now, the cut out gene of interest from the source DNA and the cut vector are mixed and ligase is added. मानने कौन आ रहा देखो, आमो पाखरे ऑलरेडी फ्रैगमेंट ही अछि, से फ्रैगमेंट रो गोटे डेफिनेट सीक्वेंस थिबो. The fragment must have a definite sequence. Now, जो particular vector DNA रे मते आको ऐड करियार अछि, से वेक्टर डीएनए रे मध्य मते सेम जगह रे गोटे कट करिया पड़िबो. व्हाई? बिकॉज सेही सीक्वेंस टा जो जगह रे थिबो सेहीटी ये रिप्लेस हे परिबो ना. व्हाई? बिकॉज जो डीएनए रे तमरो सीक्वेंसेस रहिबो, जे कोनसी जगह रे तुमे you cannot add it. So the sequence of this particular gene of interest and that sequence where it will be inserted in the DNA must be the same. Otherwise, it will not accept it or it cannot be added to the DNA. बुझ पालो कथड़ा? तो आमे कौन करिवा ना प्रथमे से जो restriction enzyme use करथिला आमे to cut out the desired gene of interest, same no restriction enzyme को use करके आमे एटी एई particular DNA रे मध्य cut टी करिवा using the same particular restriction endonuclease. What we'll do is we'll cut at the same sequence cut करके, then only we will obtain the site where that particular gene of interest can be added. Okay, and then once cut is done, a cut किए खोला restriction endonuclease. Okay, तापरे once एटा हे गला परे then जोड़ीबा using ligase. ठीक है, and once that gene of interest is now added to this DNA, ये हे गला तमोरा recombinant DNA. So एही वेरे प्रथमे हमें recombinant DNA तैयारी कर ले. Now ध्यान दीजिए थोड़ा यहाँ पे समझ में आएगा. This is the vector DNA. एटा ला gene of interest. Okay, तो एही जिनसटा खाली हेला केमती काहेकि ना ओरिजिनली जो ये थिला एमती है कि ठीक अछि आमे तो एही gene of interest रो सीक्वेंस जानी छे. तो एठी कोन कले ना आमे इंसर्ट कले गोटे पर्टिकुलर रेस्ट्रिक्शन एंजाइम. एही रेस्ट्रिक्शन एंजाइम टा कोन कला एही पर्टिकुलर पोजीशन रे ताको काटी देला. रेस्ट्रिक्शन एंजाइम टी एही पर्टिकुलर पोजीशन रे ताको काटी देला कागो ना वेक्टर डीएनए को. Once it has cut the vector DNA, ठीक है, now the sequence is same. एपट a gene of interest and एपट a vector of no that particular sequence where it is cut are the same. So the gene of interest can now be easily inserted or added at that location. A addition by कौन कले हमें ना enzyme ligase use कले. What kind of ligase it is? DNA ligase. Ligase माने जोड़ीवा. So हमने DNA ligase जोड़ के डाल के that gene of interest and the original vector DNA को जोड़ी दिलु. Now which is this thing that is obtained, the structure that is obtained, it is referred to as recombinant DNA by rDNA. In this way finally हमको recombinant DNA मिल गया. Okay, अब इसमें है क्या? This is the vector DNA. This is the vector DNA and it contains the gene of interest. ठीक है, तो एबे पर्यंत हमें कौन कले आइसोलेशन कले. ठीक अछि तार जेनेटिक मटेरियल रो. तापर हमें कौन कले एक्सट्रैक्शन कले. एक्सट्रैक्शन कार जीन ऑफ इंटरेस्ट रो एंड साइमल्टेनियसली फ्रैगमेंट्स तैयारी करकी जेल इलेक्ट्रोफोरेसिस करकी we have also obtained the gene of interest. Fine. Say gene of interest ko taapore again we also have isolated and then inserted into the vector DNA to obtain recombinant DNA. ठीक अछि, तो एही पर्यंत हम काम सरला. एबे जो recombinant DNA टी हमें फॉर्म कले, जो recombinant DNA टी हमें फॉर्म कले, that particular recombinant DNA can also be increased in number or amplified in number. ठीक है, fine. But before that, same जिनसटा तो हबो आमर actually कौन है gene of interest को multiply करिवा. Gene of interest को multiply करिवा. तो PCR बा polymerase chain reaction, PCR बा polymerase chain reaction helps us in amplification. Okay, is the synthesis of multiple copies of the gene of interest in vitro. In vitro, matlab within it, using two sets of primers and the enzyme DNA polymerase. Two set of primers, kuda RNA primers and the enzyme DNA polymerase. ठीक है? Understood? So primers and DNA polymerase use करके हमें polymerase chain reaction रा काम करिबा. Clear? Primers are small chemically synthesized oligonucleotides. ठीक है? Primers are small chemically synthesized oligonucleotides that are complementary to the regions of the DNA. तो जो DNA हमें वेक्टर हिसाब रे यूज कर ले सेही जीन ऑफ इंटरेस्ट तार मध्य अछंती and are complementary to it, as a result of which easily match करि परिबो and हमें easily insert मध्य करि परिबो. ठीक है? But the primary objective here is not that. The primary objective here is in order to multiply in large numbers, ठीक है? The DNA polymerase as well as the primers, they also have to act fast. ठीक है? बुझिएला? तमे एई जिंसटी निश्चय पढिथिबो in case of the plasmids. Yes, plasmid case re you will have the similar kind of concept. How? Now, processes of recombinant DNA technology में amplification of genes of interest using PCR. Now, how is this done? Amplification of gene of interest using PCR. How is it done? ठीक है, ध्यान दीजिए पहले. See, as you can see, here we are focusing upon the amplification part. Now, if you are focusing upon the amplification part, that particular site or region which has to be amplified. Okay? That must be amplified. Okay? So, our particular gene sequence ta koi hame focus kariba. Emiti jodi replicate habo, so sabu genes are replicate habo na. It is not that you only specifically a sequence hi habo. Entire DNA has to be copied. Fine. How? तो यही जो जिनसट वाला रीजन टू बी एम्पलीफाइड पहले डिटरमाइन हो गया. फिर इन दैट मीन टाइम दिस इज डबल स्ट्रैंडेड डीएनए. ठीक है, तो प्रथमे हमको ए जिनसटा को डीनेचुरेशन करे पड़ियो, दैट इज ब्रेक डाउन. ठीक है, नाउ वंस डीनेचुरेशन इज डन, देन हुआ क्या, दे हैव नाउ सेपरेटेड. ओके, denaturation sangare heat diya gala. Because of that, ye 5' to 3', 3' to 5', what happened to them? They got separated. Okay, alog aahi gale. And this mechanism is referred to as annealing. ठीक है, annealing. उसके बाद देखिए, the next thing that you see is the use of certain kinds of enzymes to promote the process. Why? Because the polymerase which is present, that is DNA polymerase, is stacked polymerase, thermus aquaticus. ठीक है, that is stacked. Stack क्या है? Basically polymerase enzyme है. Stack is obtained from the thermoacidophile known thermus aquaticus. ठीक है, thermus aquaticus mostly found in volcanoes and deep sea. ठीक है जी. So DNA polymerase, Taq polymerase, deoxynucleotides, all these are primarily into the amplification pathway. ठीक है? So approximately a condition रे, no, the extension has to happen. Extension matlab that particular no, or 5 prime to 3 prime chain. Fine. So sethere nua no nucleotides mane formation है पे accumulate है. Yes, that is referred to as extension. ठीक है. कैसे होती है extension, ध्यान दीजिए थोड़ा. सपोज मान लो ये 5 प्राइम टू 3 प्राइम है. ठीक है. ठीक है. So here we have the complementary pairs. ठीक. Okay. Now when we have these complementary pairs, as the movement occurs, तो AT भाभी प्राइमरी लगेगा. ओके. एंड यहां तक आना है आपको. ठीक है जी. So what happens because of this? Once the primary is attached and the process has to move, this is referred to as annealing. ठीक है. Fine. And this annealing, no, के बाद next thing that comes is extension. Extension मतलब लंबा होना. Yes. Formation of new bonds and new structures. Fine. वो हो गया आपका तुम्हारा extension. सात वालों को देखो 30 cycles वाला. 30 cycles के बाद the amplification is 1 to 1 billion times. 1 to 1 billion times in 30 cycles. ठीक हो जी. So basically कौन है अगला step? Denaturation, annealing and extension. Denaturation, annealing, extension. Clear? Oh. What are the steps of the PCR? प्रथमे denaturation. ठीक है. उसके बाद annealing and finally extension. Clear? Okay. तो ये एक अलग steps of PCR, that is denaturation, annealing and extension. अब देखिए, the amplification of the gene of interest using PR. The polymerase chain reaction is the synthesis of multiple copies of the gene of interest in vitro using two sets of primers and the enzymes DNA polymerase. Primers are small chemically synthesized oligonucleotides that are complementary to the regions of the DNA. We already know that. ठीक है. तो सही थी भाई, तो सही जगह रही लगी परी में. ठीक है. ठीक है. ओहो सॉरी. तो प्रथम में first step, what is denaturation? Just go through it once and then we'll discuss. Quickly. Denaturation की कौन ना देखो फटाफट. Okay. Now look into denaturation here. Denaturation word माने हैला भांगी दवा, break down, and because of which CL तार original condition or structure change है गला, बा original structure how effective रोहिला नी. That is known as denaturation. Clear? So denaturation basically implies the process by which the structure is broken down in order to no bring certain changes, modify anything. So यही जगह रे कौन होची? Heating of the target DNA. तो जिंसटी को heat करा गला. As a result of which it will separate. Fine. The double stranded structure now separates. Temperature कौन थिला on an average? Approximately 94 degrees Celsius. And over a span of 10 to 15 minutes approximately. Then each strand acts as template for DNA synthesis. Each strand acts as a template for the DNA synthesis. ठीक है. उसके बाद देखो एनिलिंग जमती हमर डिनेचुरेशन करिके हमें सेपरेट कर देथिले वैसे ही आपकी एनिलिंग में होता क्या है से 94 डिग्रीज टी को कमेगी 52 डिग्रीज पे ले आया 94 degrees Celsius is now reduced to 52 degrees Celsius. Because of this, at the 3' end of the DNA, ठीक है, दो primers आपकी जुड़ गए. See here, it is the joining of the two primers at 52 degrees Celsius. So at 3' end, I have the DNA templates. ठीक है. Joining of the two primers at the 3' end of the DNA templates. ठीक है. Fine. This is known as annealing. जेमती denaturation अलग ହେଇଥିଲା ଏଇଠି କଣ ହେଇଯିବ ନା ପୁଣି ମିଶିବେ. Okay? Because of the two primers. Now, then comes extension. Extension ଯେମତି ନାଟି ବୁଝି ପାରୁଥିବ ସେଇଥିରେ. What is that? Extension implies elongation of the chain. Elongation of the chain. Now, it is the addition of nucleotides to the primer using a thermostable DNA polymerase called Taq polymerase. Thermostable DNA polymerase called the Taq polymerase. It is isolated from a bacterium, Thermus aquaticus, remains active even at high temperatures during the denaturation of double-stranded DNA. बुझ पा लो? तो प्रथम denaturation करा गला, ठीक है. उसके बाद annealing करवा दिया, and finally the extension. ठीक है जी. Right? Through continuous replication, the DNA segment is amplified up to 1 billion copies. Through continuous replication, the DNA segment is amplified up to 1 billion copies. The amplified fragment can be used to ligate with a vector for further cloning. माने कौन? आमे इतने एक जिंसटी extension करी की तैयारी कर देले, okay? तो सेही जिंसटी को और सेमती न रखी की तारा amplification कर देले. The amplified fragment can be used to ligate with a vector for further cloning. ठीक है? Fine. That is what is called extension. Now, ये basically आपकी processes of denaturation, ठीक है, annealing and extension है. Denaturing में कुछ नहीं है, annealing में बीच में घुस गया, and extension रे duty separate form में गए. Okay, fine. The very simple thing, polymerase chain reaction की पहली step में denaturation हो गई, क्यों? Because of the 94-95 degree Celsius temperature. तब सही insert कॉमी की 30-35 के आसपास हो गया, that is annealing, join नहीं होला उतरे. Again, when the new strand is synthesized, then also the formation was appropriately done. ठीक है? Then insertion of the vector DNA into the host cell. Using any methods, the ligated DNA is introduced into recipient host cells. ठीक है? They take up DNA from its surrounding. Clear? So using any method, the ligated DNA is introduced into the recipients, that is the host, to grow and flourish inside it. Clear? Hence, they take up DNA from its surrounding. Now, if a recombinant DNA bearing ampicillin resistant gene, ठीक है, ampicillin resistant gene, recombinant DNA bearing ampicillin resistant gene, कैसे हो गया? Eta thorabinios a plasmid T, ठीक है? Now, in that particular state, ठीक है, that particular state A करे. So if you remember the vector, that is the आपकी bacterial plasmid, तो मोनोथियो तमरो से PBR 322 तो सेटी A साइड रे तमरो टेट्रासाइक्लिन रेजिस्टेंट, A साइड रे एम्पिसिलिन रेजिस्टेंट, ऐसा करके है. ओके, तो सही इंसर्ट ही एक्टिवेट अब फाइन. So if a recombinant DNA bearing ampicillin resistant gene is transferred into E. coli cells, the host cells become ampicillin resistant cells. ठीक है? Fine. Now, if the transformed cells are spread on the agar plates containing ampicillin, simple सी बात है, जेहेतु एबे ampicillin resistant को हमें कौन कले तैयारी कले, तो एबे यदि हमें बैक्टीरिया को से कॉलोनी रखिबा and ampicillin मिसेइबा, ताहले मध्य there will be still the bacterial colony as alive. किंतु यदि ampicillin resistant gene टी नहीं, तापरे ampicillin पके देले medium रे, तो the entire particular colony will be dead. बुझिएला कथाटा? So if the transformed cells are spread on agar plates containing ampicillin, only transformants will grow. काहे कि जो माने recombinant हे जाई छथि, जानको भितरे से ampicillin resistant gene डी पळे आसी, से माने केवल सर्वाइव करि परिबे कागु ampicillin को, remaining जो थिबे से तांको affected हे जिबे, and this implies they will be killed or they will die. Which parlo काहे कि because ampicillin is an antibiotic. Okay. So eighth रे जो माने survive करिगले, those who have survived are known as transformants, and those who do not are referred to as non-transformants. Clear? Fine. उसके बाद obtaining the foreign gene product. Now when you say foreign gene product, what do you mean by this? Once the process is completed, mane that recombinant DNA is formed and it has been introduced into the host. So the host now manufactures or utilizes that particular gene to manufacture something. ठीक है? वही चीजें देखो यहाँ पे. The ultimate aim of recombination the ultimate aim of recombination in recombinant DNA technology is to produce a desirable product, a protein. ठीक है हमें इतने यात्रा कौन पाई करचे because it ultimately to result in the formation of a desirable protein जोड़ा हम दौर कर ठीक है if a protein encoding foreign gene is expressed in a heterologous host ठीक है it is called a recombinant protein. Now what is heterologous? ठीक है if a protein encoding foreign gene is expressed in a heterologous host, it is called a recombinant protein. Kaigi? Because it was there. Okay? But in a non-encoded form, hence we have we have obtained the protein from that particular host, and now we are looking at extension bar, removal of it. Okay? How? अच्छा रे देखो फटाफट cells with the foreign genes can be grown in laboratory. ठीक है the cultures are used to extract desired protein, purify it by using separation techniques. ठीक है खाली substance obtain कले हबनी but ताकू purify करिया पाई की तमरो separate करिया पाई की techniques मध्य use करिया पड़ियो. ठीक है तो प्रथमे recombinant तैयारी कले ताकू वेक्टर साहाय्य रे पठेई देले होस्ट भितर पुणी हमर आरंभ होला. ठीक है दी प्रोसेस राइट तो जितना हुआ वो ट्रांसफॉर्मेंट्स है जितना नहीं हुआ वो नॉन ट्रांसफॉर्मेंट्स है. ठीक है cells can also be multiplied in a continuous culture system. ठीक है here the used medium drained out from one side, fresh medium is added from the other side. ठीक है it maintains the cells more physiologically active, so produces a larger biomass. It yields more desired proteins. Now very important thing एटी कौन है ना continuous culture system ने मध्य जिनसे करी हबो. ठीक है now it maintains the cells more physiologically active and so produces larger biomass. Can continuously multiply करते increase करिए volume रे as well as in number, hence larger biomass. ठीक है now how do you obtain the product which has been manufactured in the host? ध्यान से देखिए बायोरिएक्टर को quickly and then we'll move to the details. जल्दी okay now see this bioreactors age insert देखो जो तमें these are what we call bioreactors. These are large tanks. ठीक है large tanks with several equipments etc added to it. ठीक है आंख भीतर हुए कौन those host cells normally bacterial forms ला सेमानंकु आमे यूज करिबा एठी by developing a culture system. पूर्व जो culture system पढ़ना थी लगी इससे थोड़ी से पहले जस्ट पिछली स्लाइड में हमने कल्चरल देखा क्या कंटिन्यूअस कल्चर सिस्टम सेही जिनसे को ही यूज करिबा हमें कोठी in case of bioreactors. Now look at this. These are the vessels or containers in which raw materials are biologically converted into specific products. Raw materials are biologically converted into specific products, that is enzymes etc using microbial that is bacterial forms of plants, animals or even human cells. ठीक है bioreactors are used to produce large quantities of products. They can process 100 to 1000 liters of culture. The bioreactors can be very small as well as very big. Limitation रे 100 liters ରୁ ନେଇକି 1000 liters ପର୍ଯ୍ୟନ୍ତ it can perform. ठीक है? A bioreactor provides optimal growth conditions. Why? Because सब जिनिस control form रे रहला. Temperature भी बहुत अधिक नहीं, pH भी बहुत कम बेसी नहीं, substrate भी ज्यादा है, salts and vitamins etc. oxygen etc. also present. ठीक है? Fine. So bioreactor provides optimal growth conditions. Temperature, pH, substrate, salts and vitamins or oxygen to get desired product. Clear? इसमें कुछ it's not a problem. Fine. Okay. Now, as you can see in these given pictures, we see two different kinds or forms of bioreactors. Okay. Now, the first one is simple stirred tank bioreactor. Then, second, sparged stirred tank. Stirred tank. मतलब sparged है, bubbles चुगु, stirred होची, ठीक है, and tank भी है, ठीक है. Then simple stirred tank bioreactor and sparged stirred tank bioreactor through which sterile air bubbles are sparged. बस छोड़ा जाता है, ठीक है. Most commonly used bioreactors are for stirring type. मन रखियो. Stirring type, simple stirred type. Okay. It is usually cylindrical or with a curved base due to the to facilitate the mixing of the reactor contents. Okay. The stirrer facilitates even mixing and oxygen availability. Alternately, air can be bubbled through the reactor. ठीक है? Okay. Now, what are the various parts involved in a bioreactor? Look into it. पहला एक agitator system है. Agitator माने जे भूल ले वो. The motor system. This thing which is rotating the entire internal structure is known as agitator. ठीक है? फिर उसके बाद है oxygen delivery system. O2 के में जो a foam control system. ठीक है? Whatever foam is being formed, fine. तो तेरा को आया foam control system. उसके बाद देखो a temperature control system, pH control system, sampling ports for periodic withdrawal of the culture. ठीक है? ए sampling ports मतलब whatever has been achieved by the help of the bioreactor, तो उसमें क्या होता है, towards the bottom there is a releasing cap. Is a releasing cap. Now that releasing cap, ठीक है, it does not take sampling ports for periodic withdrawal of the culture. तो जितने बैच प्रोसेसिंग होची इटा आजी पड़ला इटा काली की बाहरी हो. Again जैसे वो निकलेगा दूसरा घुसेगा. So this is a continuous overlapping process. बुझेला रे. Hence अंक मित्र भलोटा कुटा the stirred type. Sparge केसरे कौन देले extra ना bubbles from the inside. ठीक है? Finally come to the downstream processing. Downstream processing का अर्थ होता है, ठीक है, डाउनस्ट्रीम प्रोसेसिंग का क्या मतलब होता है? डाउन का मतलब डिसेंडिंग. यस, ठीक है. तो एक ही देखो तो मैं अपस्ट्रीम प्रोसेसिंग इज मूविंग हेड टुवर्ड्स द मॉर्निंग. ठीक है. नाउ देखो इधर downstream processing and upstream processing जहाँ भी तो our simple objective is to obtain the product. Yes. How do we obtain the product? देखो इधर, it's a series of processes such as separation, purification of products, ठीक है, after the biosynthetic stage. Clear? Understood? Series of processes such as separation and purification of products after the biosynthetic phase. The product is formulated with suitable preservatives such that formulation undergoes clinical trials and strict quality control. ठीक है? बुझेला रे? तो एटी बेसिकली होउची कोन मल्टीपल स्टेप्स यूज करकी आमे अलाउ करछे से बैक्टीरिया एटसेट्रा को ता काम करियर. Once the work is done, then it goes for the storage purposes. ठीक है? Fine. तो एमती करे कि we have now finished the processes in biotechnology. ठीक है? Thank you people. Thank you very much. Hope you would have understood the thing. Suppose anybody has any doubts regarding the biotechnology principal processes. ठीक है? You are always welcome to ask the questions. ठीक है? चलो बच्चा लोग, take care, enjoy your day. ठीक है? पढ़ाई वढ़ाई ठीक से करना, खाना पीना ठीक से खा लीजिएगा. Thank you very much बच्चा. चलो, have a nice day.